welcome back to Sachin Biology classes. And in this session we are studying about double fertilization. The name very clearly it explains about the meaning. Two times fertilization ಆಗುತ್ತೆ, ಅದಕ್ಕೆ ನಾವು double fertilization ಅಂತ ಕರೀತೀವಿ. Let us discuss in detail that how two times fertilization it takes place in embryo sac. Okay. When the pollen tube which is passing through any one of these synergid cell, ಯಾವುದಾದ್ರೂ ಒಂದು synergid cell ಮೂಲಕ pollen tube ಎಂಟ್ರಿ ಆದಾಗ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಒಳಗಡೆ pollen tube ಒಳಗಡೆ ಇರತಕ್ಕಂಥ ಕಂಟೆಂಟ್ಸ್ ಯಾವ್ದು they are nothing but two male gametes and they releases into the cytoplasm of synergid cells. Okay, and after releasing the gametes in the cytoplasm of the synergid cells and one male gamete, it moves towards the egg cell. Okay, so here are the two male gametes. They released into the synergid of the synergid cells, and one male gamete it fuses with egg cell and it results into the formation of a diploid zygote. And next, the another male gamete which was there, that is moves towards the polar nuclei and fertilizes here, and it forms the triploid primary endosperm nucleus. Okay, and what is the fate of this triploid primary endosperm? Fate ಅಂದ್ರೆ ಅದು ಏನಾಗುತ್ತೆ ನೆಕ್ಸ್ಟ್? ಒಂದು ಮೇಲ್ ಗ್ಯಾಮೆಟು ಎಗ್ ಸೆಲ್ ಜೊತೆಗೆ ಫ್ಯೂಸ್ ಆಯಿತು, ಅದು ಎಂಬ್ರಿಯೋ ಆಗಿ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಇನ್ನೊಂದು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಏನಾಯಿತು, ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ ಜೊತೆಗೆ ಫ್ಯೂಸ್ ಆಯಿತು, ಅದು ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಅಂತ ಕರೆದ್ವಿ. ಟ್ರಿಪ್ಲಾಯ್ಡ್ ಅಂದರೆ ಇಟ್ ಇಸ್ ನಥಿಂಗ್ ಬಟ್ 3N ಕಂಡೀಷನ್. Okay, hence we are calling that 2N condition. When one male gamete it fuses with that, it becomes a triploid. So it is primary endosperm nucleus is a 3N condition. Later it becomes a primary endosperm nucleus. Then simply we used to call as pen anta karitevi. What is that? Simply we used to call as pen. That is nothing but primary endosperm nucleus. Then what is the function of this pen? So later it develops into an endosperm. Endosperm, which is consisting of rich reserve food material and which is very essential to the development of embryo sac. In the post-fertilization events, and post-fertilization events are nothing but after fertilization and events of endosperm and embryo development and maturation of ovules into seeds and ovary into fruit. And all these three events are collectively termed as a post-fertilization events. And in this post-fertilization events, it forms three structures, and those three structures are nothing but an endosperm, embryo and seed. And let us discuss one by one about the structures. The first one is an endosperm. What is endosperm and how the endosperm it forms? As we already know, where in an embryo sac there are polar nuclei, two haploid nucleus that you can see, that is N plus N. Okay, and these two polar nuclei they are fused with the male gamete, so which is produced by a pollen grain. During the fertilization process, that pollen tube releases the two male gametes inside the embryo sac. One male gamete is fused with the egg cell and it forms the diploid zygote, and another male gamete that is fusing with the polar nuclei, and see here one nucleus and here two nucleus, and all these nucleus fuse together, it forms a large nucleus, and that is nothing but a primary endosperm nucleus. What is the difference between primary endosperm cell and primary endosperm nucleus? The primary endosperm nucleus which is formed within an embryo sac and completely we are considering that as a cell. What is a primary endosperm cell? And this within this cell there is a developing zygote is also there. Okay, and this zygote later it develops into embryo, and this primary endosperm cell and undergo the division and it forms a triploid primary endosperm. Primary endosperm tissue which is consisting of abundant reserve food material, and this reserve food material is used for the developing embryo. Okay, and let us discuss that once the primary endosperm nucleus is formed, it undergoes the successive nuclear divisions to give rise to free nuclei. And what does it mean of free nuclei? As we already studied in first year, that is karyokinesis and cytokinesis. Karyo is nothing but nucleus and kinesis is nothing but movement. So we are studying in cell division here. And only nucleus undergo division, not a cell or cytoplasm. So hence only a karyokinesis takes place and produces thousands of nuclei. And subsequently the cell wall formation occurs and endosperm formation it takes place. And how that endosperm becomes cellular, just you look at this diagram, you can understand completely. And this is what the structure of endosperm, and this is what an embryo, where embryo ke development ಆಗಬೇಕಾಗಿದ್ರೆ ಇದಕ್ಕೊಂದಿಷ್ಟು nourishment ಬೇಕಾಗುತ್ತೆ. Then who is providing the nourishment? That endosperm is providing the nourishment. So how? Where the primary endosperm nucleus which is undergoes successive nuclear divisions to give rise to free nuclei, and all the free nuclei which are produced by the cell division, they start to push towards the periphery region because of the large vacuole. And later what it happens, all the free nuclei which are moved to the periphery region and they start to form a cell wall. You can see this is the reason where the cell wall formation takes place, and this later it becomes a cellular endosperm. Okay, and you can look at the diagram here. This is what the coconut diagram, and the best example for free nuclear or nuclear division ಗೆ ಒಂದು ಬೆಸ್ಟ್ ಎಕ್ಸಾಂಪಲ್. ಯಾಕಂದರೆ ನಾವು ವಿಧವಿಧವಾಗಿರ್ತಕ್ಕಂಥ ಅಥವಾ ಬೇರೆ ಟೈಪ್ಸ್ಗಳನ್ನು ನಾವು ಕಲಿಬೋದು ಎಂಡೋಸ್ಪರ್ಮಲ್ಲಿ. ನ್ಯೂಕ್ಲಿಯರ್ ಎಂಡೋಸ್ಪರ್ಮ್, ಸೆಲ್ಯುಲರ್ ಎಂಡೋಸ್ಪರ್ಮ್, ಹೆಲೋಬಿಯಲ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಅಂತ ಟೈಪ್ಸ್ ಇದೆ. ಬಟ್ ಆ್ಯಸ್ ಎನ್ ಸಿ ಆರ್ ಟಿ ನಾವು ಓನ್ಲಿ ನ್ಯೂಕ್ಲಿಯರ್ ಎಂಡೋಸ್ಪರ್ಮ್ನ ಡಿಸ್ಕಸ್ ಮಾಡ್ತಿದ್ದೀವಿ. ಸೊ ಅಕಾರ್ಡಿಂಗ್ ಟು ಯುವರ್ ಸಿಲೆಬಸ್, just you look at this diagram. This is the section view of, okay, section view of coconut, and you can see the green color outer part, that is nothing but an epicarp anta kariತೀವಿ, and in between, so next to the epicarp there is a fibrous tissue, and that fibrous tissue is called what? A mesocarp anta kariತೀವಿ. And then inside the brown color layer, that is nothing but an endocarp anta kariತೀವಿ. Inside the endocarp there is a white kernel, so that is edible part, that is called endosperm. Okay, so that is an edible part. It is nothing but a kernel anta kariತೀವಿ. And how it forms that? Even you can see within the coconut, so you can see the milky fluid. So in milky fluid, ನಾವು ಇದನ್ನ ಎಲ್ಲಿ coconut ಅಂತ ಕನ್ಸಿಡರ್ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ಎಲ್ಲಿ ಫ್ರೀ ನ್ಯೂಕ್ಲಿಯೈ ಇರ್ತಾವಲ್ಲ ಆ ಫ್ರೀ ನ್ಯೂಕ್ಲಿಯೈ ಎಲ್ಲ before cellularization. Please try to understand, before cellularization is nothing but before the formation of cell wall. Okay, again formation ಮಾಡಿರುತ್ತೆ, it forms a liquid, and this is nothing but a milky liquid ಅಂತ ಕರಿತೀವಿ. ಈ milky liquid ಒಳಗಡೆ ಫಾರ್ಮ್ ಆಗಿರುತ್ತಲ್ಲ ಅಲ್ಲಿ ಯಾವುದೇ ರೀತಿಯ ಫ್ರೀ ನ್ಯೂಕ್ಲಿಯೈಗಳು ಇರೋದಿಲ್ಲ. ಲೇಟರ್ ಏನಾಗುತ್ತೆ ಅಂದರೆ they are starting move towards the periphery and it forms a cellular endosperm kernel. Cellular endosperm, that is what? An edible part of the fruit. And this is what about an endosperm. And sometimes we feel it's very easy concept, but it creates a bit of confusions in students while studying. So if do you get any doubts, drop your comments in comment section. I want to stop this session here and we can continue. the next session with a new concept. Until that, take care, and always you please remember that study for knowledge, not for marks. Until that, take care.